let's get into communicable diseases. Quite a lot of stuff in this topic, and I'm going to try and do it as quickly as possible. Remember, the idea is not that I'm going to try and re-explain everything to you, but the point is going to be to kind of make sure to organize all that content, to give it some kind of structure, and just to point you in the direction of some key things to ensure that you revise those things so that you're not missing anything out before the big day. Okay? Right, let's get into it. It's difficult to organize this one because it is all very, very highly connected, and kind of one thing bleeds into the next. But the three main areas that I've divided this into is the first part looking at the pathogens, which are the agents that cause communicable disease. Okay, and in that we're talking about all the plant pathogens, the animal pathogens, the different types of pathogens, the virus, prototists, bacteria, fungi, and those examples. All right, and in that one I've also included the transmission and the factors which affect transmission. So that's one big area. The other very big area is defense. So the defense, basically we've got the pathogens, then we've got the organisms that have been or potentially will be infected by the pathogens, and the defense mechanisms that those organisms have against the pathogens. So we're going to look at plant, animal. Plant defenses are relatively straightforward, and we need to know how they work and the different examples. In animal defenses, we kind of go in a process. We've got all the non-specific defenses which essentially are preventing the infection by the pathogen, and then we look at the specific immune response that being made, I guess, when the infection has occurred. So the pathogen has bypassed, or not bypassed, but got through somehow the non-specific defenses, and now within the blood, within the tissues, within the cells, the defenses will now try to remove that pathogen from the body, and how that happens. This kind of leads into immunity and the types of immunity, and then that takes us to vaccination, which is immunity, kind of an induced, an artificial kind of immunity. And vaccination is then in this big area of disease prevention. Okay, so kind of treatments for diseases. So vaccination is kind of like a prevention, but we can also treat diseases, so this should be really disease prevention and treatment. Treatments then have medicine, or treatments are treatments. So treatments include medicines, and medicines can have different sources and their advantages and different disadvantages. And then of all the different types of medicines we can have, we focus on antibiotics, and how there is this phenomenon of antibiotic resistance, and that takes us to the end of the topic. All right, so with that done, let's get into the detail of this stuff. All right. So we begin with communicable diseases and pathogens. Now, before we get into the types of pathogens, I think it's important to spend a little bit of time just understanding because we have, part of our body is microorganisms. We have microorganisms on the skin, we have microorganisms in our digestive system. So what makes some microorganisms pathogenic and others we are able to live with, right? So why do these pathogens cause harm? And there are a number of ways that we need to be aware of. One is that they take over the cell metabolism. So viruses enter the cell, and the cell starts translating or transcribing and translating, i.e. expressing viral genes to reproduce the viral particles, but that takes up the cell's energy and resources. So all the cell's energy and resources are diverted to make more viral particles, which then leave the cell, kill the cell, leave the cell, and then cause infection in neighboring cells, and the same process is repeated, and this causes lots of cell death. Tissues then, don't function properly, organs don't function properly. Okay, another way is simply by causing cell death. So while viruses take over the metabolism, so they're kind of invading into the cell, inserting their genetic material into the cell and that stuff, the other kinds of microorganisms don't necessarily do that, but they do cause the cells to die, and I guess use the biological material from those dead cells to fuel their own cell division, reproduction, advance their life cycle, etc. Okay, and also some microorganisms will release toxins and as part of their normal life cycle, but they're releasing toxins which will cause harm to the host organism, eventually may cause its death as well, because these toxins interfere, block, bind to, prevent from working properly the host enzymes, proteins, and because the host enzymes, proteins don't work properly, this causes lack of cell function, potentially cell death. It disrupts the host organism's functioning, basically. All right. Okay, so we just need to be aware of how these things can happen. All right, next, we go to types. So the types of pathogens then, okay, so the way I've divided it is we've got the plant pathogens, so the pathogens that cause diseases in plants, and we've got the animal pathogens. All right, so that's our first division, okay, but for each of these there are the different types of pathogens. So we've got plant pathogens, so pathogens that infect plants. Now of the pathogens that infect plants, we do have some different types. Okay, now do need to remind myself of this because I'm not very good at remembering this. So in terms of plants, we have the 4 kingdom groups. So we've got Protoctists. Okay, now the example of the Protoctist plant pathogen is potato blight. Okay, so the disease is the potato blight that's caused by a Protoctist. Okay, then we have bacteria, so we've got a bacterial pathogen, bacterial plant pathogen. Now, in the case of the bacterial plant pathogen, we have ring rot. Okay, now then, so we've got Protoctist, bacteria, we should have fungus or a fungi. Now, in that case, we've got black sigatoka. Okay, and the plant pathogen that itself is a virus is tobacco mosaic virus. Okay, right, and then the same thing with the animal. In terms of animal, we've got 2 types. We've got human ones, so human specific, and then we've got general kind of animal or pathogens that infect animals more generally. Okay. Okay, so that's how we'll do it. So in terms of the human specific ones, we have a virus, right, which is HIV, and that causes AIDS. All right, and we then have fungus or fungi, and that one is athlete's foot. Okay, we have 2 examples of bacteria in humans. So we've got bacteria, 2 examples. One is TB and the other one is bacterial meningitis. Right, and I think we don't have protoctist. We do though, don't we? Yeah, okay. And then general animal ones. So in terms of the general animal ones, I'm going to put the protoctist here. So protoctist, I'm going to put protoctist here as well. So actually, let's do that. Right, protoctist. So the protoctist example is malaria. So the pathogen that causes malaria is a protoctist. We can just about see that, hopefully. Right, now the pathogen that causes malaria is a protoctist, but as part of its life cycle, interestingly, it does infect mosquitoes, and part of its life cycle is in mosquitoes, so it's infected mosquitoes, but another part of its life cycle is in humans, in the red blood cells. So we have the protoctist as part of the animal and human categories. Okay, and then animals more generally, we have influenza as a virus. So animals generally, we have virus, and that will be HIV. And animals generally, we have a fungus or a fungi, and that is ringworm. Okay, so I'm just compartmentalizing things as best I can, and there we have all the plant and animal pathogens. And then the next thing that takes us to is transmission. Okay, so in terms of transmission, what I would point you to is and look here, before we go any further, with the pathogens is an ideal opportunity for examiners to test your understanding of 2 things. One, cell structure. Okay, so I'm just going to make a note here. There's other topics that this connects to. One is cell structure, because you're looking at protoctists, you're looking at fungi, and bacteria, which are cell-based organisms. So you would need to be aware of what features they have. Again, this would connect to, so be aware of which organelles which organelles and structures you would expect to be present in a fungi versus a protoctist versus a bacterium. It's also an ideal opportunity for an examiner to maybe test your understanding of classification. Okay, now obviously not in depth, but at least from a level of being able to identify the different kingdoms. Right, we've got prokaryotes, we've got fungi, virus, and bacteria. Right, so I guess in terms of cell structure, prokaryotes versus eukaryotes. In terms of classification, you've got these different kingdoms. Would you be able to look at any given cell and be able to recognize it as a fungus, as a protoctist, as a bacterial cell? Right? Okay, I think we'll leave that there. Let's move on to transmission. In terms of transmission, pretty straightforward. The first kind of categorization that I've got are the types of transmission. Okay, so remember, in terms of types, we've got direct and we've got indirect. Basically this is the story of the pathogen, but how do these pathogens get from one host organism to the next host organism? Because that's the only way the pathogen's life cycle can continue. Okay, It's tied up with its reproduction, moving from one host to the next. Okay, so types, we've got direct and indirect, right? In terms of direct and indirect, how I would subdivide it is know the examples of direct transmission in plants, know the examples of direct transmission in humans, right? Or animals, human/animals, all right? In terms of indirect, the same thing, like, yeah. So what is an example of an indirect mechanism of transmission for plants, for example? And that might be something like water, which is not via direct contact, but the pathogen would move from one host via the water to the next. That would be indirect. So know the examples that you might have in each case for each different type of organism. Plants, human/general animals. Okay, yep, and yep. For each, know the examples. Okay, right, now that that's done though, the last thing is factors. So factors is another layer on top of all this. It's not like a separate thing. So in terms of transmission, so these direct mechanisms or indirect mechanisms of transmission, they can be affected by certain factors, right? So if we've got like indirect transmission in animals, that could be, for example, droplet infection. So droplet infection would be there, but there are certain factors which make droplet infection more or less likely, right? For example, overcrowding would increase the chance of droplet infection because there's simply more people in a particular space, right? Yeah. Temperature, for example. Now high temperatures might disperse these droplets more quickly, whereas low temperatures might allow these droplets to remain in the air for longer, right? So these factors are connected to direct and indirect transmission, okay? Right, so we've got factors and then we move on. So we move on then to plant defenses. I haven't left myself a lot of space there, but there's not really a lot here, I think, right? So plant defenses, I would say, it wouldn't be like your main, this is not the main part of the topic, but it obviously, like, I'm not making any assessment on or prediction about what's going to come up, what's not going to come up, what's got more chance of coming up, what's got less chance of coming up. I don't play that game. I would suggest any good student is knowing all the stuff. Okay, so in terms of plant defense, there is less depth to it. That's what I'm saying. In terms of plant defense, the two things I would point you or encourage you to know is how plant defenses work and the types, basically. Okay, how plant defenses work. Now, what I would say here, and remember that this is a synoptic paper, so I know that there's only 4 topics, but I would keep an open mind and don't be surprised or shocked when there might be a few marks here and there for a lot of non-listed topics. Now, in terms of how plant defenses work, it is very reminiscent of hormonal communication. All right, so how plant defenses work, yes, there's chemical defenses, there's physical defenses, but these there's physical defenses, I guess it's like this, isn't it? So plant defenses, we've got 2 types, right? We've got physical, right? And again, I would expect you to know the different types of physical defenses, but bark, callows, things like that, okay? No, physical defenses is simply just bark, isn't it? Yeah? Okay, so we got physical defenses, and then we have also got chemical defenses. Now, chemical defenses are very reminiscent of hormonal communication, the animal and plant responses. Basically, something happens, stimulus, right? And that stimulus causes cell signaling, and the cell signaling causes changes in gene expression. The changes in gene expression cause certain chemicals to be produced, which reduce the chance of transmission or infection of a pathogen. Okay, so it's very overlapping with that. So how plant defenses work, remember, there could be pathogen. Now, in this case, the stimulus should be the presence of the pathogen, right? The stimulus should be the antigens that are present on the pathogen. And these antigens, they might bind to some kind of cell surface receptor protein, and that causes cell signaling, and the cell signaling results in changes in gene expression, certain genes being switched on, certain genes being switched off, end result being gene expression changes causes certain chemicals to be produced. So maybe the gene that we switch on is an enzyme that produces tannins, for example, okay, or an enzyme that produces terpenoids. All right, but I don't think we need to go into too much detail on that one. All right, any question in this area will be very general. Okay, but so gene expression changes, chemical defense produced. Okay, and so all I would put down here is just have examples of the chemical defenses. Okay, so we've got our physical defenses, have examples of those. We've got chemical defenses. It's useful to know, have an idea of the mechanism by which those chemical defenses might be stimulated, produced, induced, and then have some idea of what those chemical defenses are and how they prevent or how they reduce the chance of infection, how they reduce the risk of the pathogen spreading throughout the plant or spreading to other plants. Okay, so how it works. In terms of these, a one-line explanation, perfectly fine, right? For example, chemical defenses, pathogens. Yeah, for example, callose production thickens the cell wall, making it more difficult, or reducing the chance or reducing the ability of pathogens to enter plant cells. Okay, all right. Moving on then. Oh, are we already on animal defenses? Well, that was quick. Let's keep going then. Let's not lose that momentum. Animal defenses. Let's begin. First, we begin with non-specific, and the idea here is, so actually it's an important little transition that we've made there. So we've got animal defenses, we've got plant defenses. All right, okay, so animal defenses then, the first little stop on this journey is the non-specific. Now the non-specific is all about, I say, preventing infection. Okay, the non-specific is all about preventing infection. All right, yes, and generally speaking, they all work in a kind of barrier kind of way. Yeah, so we can see them as barriers. Some are physical, some are chemical. Okay, so what are those things? So lysozyme. Now I see a lot of mix up between lysozyme and lysosomes. Lysosomes is also going to be part of our discussion, but lysozymes are extracellular enzymes that are hydrolytic, all right? So, but they're enzymes that are extracellular, and you'll find them in body fluids, okay? Lysosomes are organelles that contain hydrolytic enzymes, but they're inside cells, they're intracellular, and they're organelles, whereas this is extracellular and it's an enzyme, okay? So just be careful of that. Right, so we got lysozyme, mucus, mucous membranes. Quick note, remember that mucous membranes is also part of cells in module 2, and you know, you're supposed to be aware of the specialized function of that tissue, which we do kind of look at in this topic, so maybe not too much extra work, but just put an asterisk there. It is a synoptic paper, so we've got to be a little bit more nimble, I guess, in our approach to this paper. Right, next, skin. Right, physical barrier, dead cells, many layers, thick, preventing the entry of pathogens, and then that takes us to, why did I remove blood clotting? Oh, right, skin. So the skin is a barrier itself, but I also put in here blood clotting. Right, simply because the process of blood clotting, it kind of seals the skin, preventing the pathogens from getting in, that temporary seal, but then it also allows the wound healing to occur, right? So any break in the skin can be sealed, right? So not only does blood clotting prevent entry of pathogens into blood vessels, it also kind of allows the wound healing process to occur, sealing the skin, right? Let's move on. Then we have the next level of defense, which is still non-specific, but it is next level, and that is inflammation. In the inflammatory response, okay? So this is kind of like when the pathogens do penetrate the barriers, there is still some non-specific mechanisms in place to, to remove / destroy any pathogens that might have got through the barrier before a proper infection sets in. Okay, and maybe I would define infection as cells which are able to successfully reproduce, multiply within the body, and the inflammatory response is not dealing with it. Okay, so the non-specific response is not removing the pathogen, or is insufficient to remove the pathogen. Anyway, things get through the barriers, they could be taken care of by the inflammatory response. What is that? That is essentially cells. So mast cells can release in response to a pathogen. Now remember, pathogens are kind of, they've got their antigens on their surface and they're releasing chemicals which aren't supposed to be in tissues. So mast cells can detect that, and when they do, they can release histamine, which is a signaling molecule. Okay, now so the histamine is released and it has certain effects. Okay, it has certain effects. Somehow, yeah, that's fine, we're good. Okay, so two main effects that it has is that it causes local blood vessels to dilate. Local blood vessels dilate, meaning more blood can flow through that area. Its other effect is to make capillaries increase capillary permeability. So the gaps between the endothelial cells of the capillary kind of open up a little bit, and it allows white blood cells to escape, such as neutrophils, and allows those neutrophils to carry out phagocytosis, destroying any pathogens that might have got through that barrier. That's how it's supposed to work. Okay, which also causes fluid to be released. So increased capillary permeability, it does release neutrophils, or does allow neutrophils to enter the area and carry out phagocytosis of the pathogens. Okay, but as a side effect, it also increases swelling. Yeah, so I've seen this come up a number of times in exam questions where you are asked to explain why there's swelling and why there's redness. Yeah, so the redness because of the more blood flowing in the area, because the blood vessels are dilated. There's swelling because there's a lot more fluid in the tissue, because the capillaries have become more permeable because of the signaling caused by histamine. Okay, right, then let's move on, and that will take us nicely into the specific response, okay? Because phagocytosis is kind of like the initiating point. It's the starting point of that immune response, okay? So animal defenses, phagocytosis, how would we do this? So the phagocytosis causes, actually let's just do this like so, non-specific inflammation, but the phagocytosis allows the specific response. Let's just talk phagocytosis for a little bit, and then we'll go into the specific immune response. So remember that with phagocytosis, it's kind of like an endocytosis, like a giant endocytosis, right? And in fact, you know, that has also got then connections to cell structure, right? But phagocytosis, actually not cell structure, it's got connections to membranes, right? In membranes, we look at transport across membranes, and endocytosis was one of those mechanisms. Anyway, phagocytosis, where are we? Right, phagocytosis, you know, is all about these antigens. The antigens that are these proteins that, or carbohydrates or whatever molecule that's not really supposed to be there in the body, and the body can, you know, other cells have receptors that can bind to proteins which are not part of the self, right? So we got antigen receptor interactions. So we got on a white blood cell, on a phagocyte, like a neutrophil or a macrophage, we've got receptors. They bind to the antigens on the pathogen, right? So we've got that antigen receptor interaction, and that will cause, how did I do it here? Do receptor antigen, right? And then we have the engulfing stage. Right, so because the two are interacting, we have engulfing. So the interaction allows the phagocyte to wrap its membrane around the pathogen, right? And that's all because of the kind of regulation of the cytoskeleton. So don't forget about that, or just have an awareness of that's how that part isn't happening by magic. It's because the cytoskeleton is being reshaped. Okay, so we have engulfing caused by the cytoskeleton reshaping, and that engulfing then results in the formation. This is going to get crazy. Engulfing. Once the engulfing happens, we form a phagosome. Right, that's kind of like a vesicle inside the cell that contains the pathogen. Right, now the phagosome then fuses with lysosomes. Lysosomes, different to lysozymes, right? Lysosomes are these organelles inside the cell that contain hydrolytic enzymes. They fuse with the phagosome, forming phagolysosome, right? And from the phagolysosome, what happens is that because of those hydrolytic enzymes, the pathogen is basically broken down, destroyed. It can't cause infection anymore, okay? Because all its proteins have been broken down, carbohydrates broken down, membranes, etc. Okay, but those fragments, the bits that it's broken down to, are recycled to the surface, where now this phagocyte can act as an antigen presenting cell. Yeah? Okay? So there's a lot of stuff there, a lot of important stuff I have not put down on the board, but hopefully you've been kind of filling in the blanks yourself, right? The antigen presenting cell then is there, and the antigen presenting cell is a very important point, because from here, and I hope you can see how the non-specific responses enabled this, from here we can initiate the specific immune response, right? Because we've got an antigen presenting cell, clearly something's got through our barriers, and we need to do something about it. Cool. Antigen presenting cell, and now we get on to, I might need to move certain things around a little bit, but now we get on to the specific, I really didn't expect ending up here. We've got the specific immune response, okay? So that's another critical staging point right there, specific immune response. Let's just see how we go, and I might just end up moving some of this stuff up. Okay, right. Specific immune response. In fact, I'm just going to move that. I'm just going to get rid of that right now because I need the space. So apologies. That stuff's going to go. Hopefully I'll remember what I did there. Now let's begin with the specific immune response. So with the specific immune response, what's going to happen is our main player is the T helper cell. I don't want that to look like an M, right? Because there is such a thing as TM. But our T helper cell is the beginning part of this process. And the T helper cell, remember, is, do I really want to illustrate this? I don't think I want to do that, right? We want to stick to words because that's what we're going to be using in an exam. T helper cell. And the next important thing is, remember, we've got lots of T helper cells, but the T helper cell plus the antigen presenting cell, right, will combine and give us the process of clonal selection. The idea being, remember, that there's lots of T helper cells, each with a different receptor complementary to different antigens, but the APC, because of the antigens that it's presenting on its surface, will select one of our many T helper cells to be the one that's going to coordinate the specific immune response. So once we've got our clonal selection of the T helper cell, the activated T helper cell, right, at this point, activated, and I'm just going to put some words down, words such as complementary, specific, antigen, and receptor. These are important key terms in this explanation, right? So the T helper cell is activated. The T helper cell is now going to activate or carry out the similar kind of clonal selection of the B cell. So the T helper cell plus the B cell, T helper cell plus a B cell, and again the T helper cell will have many B cells to choose from, right? Each of these B cells presenting a different antigen, but one of these B cells is going to be expressing or presenting the same antigen as the antigen presenting cell down here. And as a result of that, it will activate and clonally select a certain B cell. So this one was a T helper cell clonal selection, and now we've just had a B cell clonal selection. Now, after we've selected the clonal the B cell clone that's going to be useful and the T helper cell clone that's going to be useful, they both undergo as a result of complementary antigen binding, so complementary binding, right? But there's going to be a release of cytokines. Again, these are signaling, because this is a cell producing something that's causing another cell to behave differently. That's cell signaling. So these are signaling molecules, right? Cytokines released, and the result of cytokines released, I think it's kind of this way actually. Cytokines released by the T cell stimulate the B cell, right? And the T helper cell was activated by binding the B cell, but we've got activated now cells, and these will undergo clonal expansion. Now, clonal expansion also includes the expansion is happening by mitosis. We're expanding the cell numbers to be able to handle the growing now pathogen numbers, because the pathogen has caused an infection. They are multiplying, so we need to multiply our immune cells, our lymphocytes, right? So clonal expansion happens, but clonal expansion is accompanied by not just mitosis, but differentiation. I am definitely going to run out of space. Okay, differentiation. Now, differentiation results in a number of different cells produced. Now, the B cells differentiate into B memory cells. Actually, let's do it slightly. Actually, yeah, whatever. B memory cells and plasma cells. The T helper cells differentiate into cytotoxic T cells or killer T cells, T memory cells, and more T helper cells, to amplify the process. Okay? Now, the important thing is that the plasma cells produce antibodies. Plasma cells produce antibodies, and the cytotoxic T cells. So the cytotoxic T cells, what they do is they kill. So any cell that's infected, kill it. Kill infected cell. Remember, infected cells will be presenting antigen on their surface, and that's how the cytotoxic T cells or killer T cells. That's how they will identify which cell needs to be killed. So that's what the T killer cells do. The antibodies do a number of things, right? So how they work is they either, they act as something called opsonins, which means they bind to the antigens on the pathogen, coat it, and attract a phagocyte, and basically target the pathogen for destruction by phagocytosis. Okay, so opsonins induce phagocytosis, but they can also cause agglutination, right, which is basically antibody binding multiple pathogens and causing them all to clump up, kind of essentially disabling the pathogen from being able to function, because they're just kind of locked in these giant pathogen balls. They can't really enter cells in that way. Okay? And also they can act as antitoxins. We said that pathogens release toxins. Antibodies can act as antitoxins because they can bind in a complementary way to the toxins. Then the toxin can't bind to the enzyme that it was going to block or disable, right? So then effectively the toxin is prevented from acting. Right, so that's antibodies, how they work. We also need to be aware of antibody structure. Again, I'm not going to go into that too much, but understand about its 4 polypeptide chains, its variable region, which is complementary to the antigen and unique to different antibodies, and its constant region, which all antibodies share, the disulfide bonds that connect the different polypeptide chains to each other, essentially giving it a quaternary structure. So, and with antibodies, there's a lot of questions that can come up regarding structure. So I'll just put an asterisk there. Antibodies are proteins, so prepare yourself to kind of know the antibody structure quite well, because it's an opportunity for an examiner to ask you about protein structure. Yeah, and look, we've had differentiation here. Again, that's a module 2 kind of topic, so you could be asked about that. We have mitosis here in expansion. Prepare yourself a little bit for that. Okay, and just like generally, look in all the topics that are on the list, look for where there's connections to other topics. All right, it is a synoptic paper, so I think we got to go the extra mile in terms of in that sense. Okay, and kind of looking through previous paper 3s, I feel their scope is broader, but sometimes there's not as much depth in paper 3. So that's kind of what I think about how you should approach this. Kind of know the biology a bit more broadly than you otherwise might, but yeah, maybe perhaps don't worry too much about the very, very specifics, details of everything. So if you have to make a decision between depth and breadth for a synoptic paper, I would definitely go breadth. Okay, right, let's move on. Let's keep going. Let's not let that ball stop rolling. Clonal selection, T helper, right. Now that is what we call a primary immune response, and that all takes time. But because of the B memory and the T memory cells, we can have something called the secondary immune response, which can be 2 things: faster and greater, faster and bigger, right? Which ultimately means fewer symptoms as a result of secondary response. Okay, now the secondary response kind of takes us to one of our next topics. Okay, antitoxin, antibodies, plasma cells, fewer symptoms. Right, last 2 things then. The idea of the secondary response, and that's something that comes up quite a lot, right? The idea, that graph that they show about the amount that, the level of antibody production in the primary response versus the secondary response much bigger. Can you explain why? Can you explain why there's no symptoms a second time or in the secondary response? So you've got to kind of call on your knowledge of the T memory and the B memory cells. Okay, right, and not having to go through the whole clonal selection and all that stuff, right? You still have to expand the populations, but you've already got these memory cells, so you don't have to go right the way from the beginning. Okay. Yeah. Cool. Let's go then. Treatments. I'm going to try and fit it into this space. Not effective use of space there. That's not very good, Dr. Bafsa. Right then. So we go. Treatments, and in treatments, right, we have the different sources. Actually, no, in treatments we've got 2 main things. We've got vaccinations, no, still not done. I guess we could go from here. We can go from antibodies to immunity, right? Because antibodies is essentially how we measure or determine if someone is immune. So the question can be asked, right, if someone has antibodies in their system, in their body, how did they get them? And that's the nature of the different types of immunity, right? So remember, if someone's got antibodies that they made themselves through an immune response, that is called active. If someone has antibodies that they didn't produce themselves, that is passive. Okay? But active can have happened in a natural way, right? Through natural processes in the life cycle of an organism. Right? But active might also have happened artificially. Artificially through a vaccine, for example. So natural might be some active natural would be an immune response. You've got infected and you made an immune response. Active artificial would be something like getting a vaccination, which is not natural, which is artificial, but you've still produced those antibodies because you made an immune response to the antigens in the vaccine. Passive, again, could be natural. You got those antibodies through breast milk, for example, and passive could also be artificial. Right? So you were injected, but not with the antigen, you were injected with antibodies. Okay, so that's how you got them. So that's immunity. Right, and immunity then takes us to vaccination programs. So active and artificial, that takes us to vaccination and the idea of herd immunity, like why do we do vaccinations and maybe the different types of vaccines that we could have. Right, am I still on the board? I think at this point it doesn't matter, everything's too small. All right, okay. Yeah, and that then takes us to treatments, like non-vaccination treatments. Because remember, vaccinations are still preventative in a way. So treatments, so we've got medicines. We need to know the sources of medicines, right? And these are not kind of anti-pathogen medicines necessarily. We're talking about medicines generally here, so things like aspirin, which is not, which is for, and it's an anti-inflammatory, but so we're talking about having an awareness for where medicines come from generally. So we could have medicines from plants, but we could have medicines from microorganisms, usually fungi, which produce antibiotics. Okay, but the other source is, I want to say, science, right? But you can get like synthetically produced drug molecules as well. You can get drug molecules designed using our knowledge of genes of disease-causing microbes, or using our knowledge of human genes to produce medicines which are more tailored specifically to work for people with certain alleles, right? So genetic information, it could be genetic modification, could be genetic synthesis. Yep. Okay, so we have those. And I think that that's it. Now, microbes produce antibiotics, right? And so there is that, but one of the disadvantages of antibiotics is, their abuse. So, maybe they're not used properly, maybe they're overprescribed, maybe on the other side the patient is not taking, or the infected person is not sticking to the prescription, the dosage, etc., or completing the course, and that can result in antibiotic resistance. Almost there. I'm going to put an asterisk there because resistance is a form, is as a result of, in a way, selection processes. So it's connected to evolution. Now, it's not a natural selection in a way because it's not nature that's selecting it, but there is a selection pressure, which is the antibiotic. There is a variation, which is the variation in the pathogen population. And there will be selection. Some of those pathogens will have a better chance of survival because of their variations. Adaptation, increased survival chances, increasing numbers of resistant individuals, bacteria, pathogens, causes the resistant population to arise. Or a greater proportion of the population is now resistant. And I think that is it. Yeah. Okay, apologies. I'm trying to kind of sped through that a little bit, but there is a lot to look at, and I'd rather that I give you guys as much time as possible to kind of get on with it. But yeah, I hope that that's kind of made some kind of story of communicable diseases. I hope it's kind of highlighted any particular areas that you might need to look at again. Yeah. All right, guys, hope I haven't missed anything out. Good luck for the last paper. Remember, it's paper 3. Remember, expect it to be a little bit more synoptic than what the advanced information says. Prepare yourself for that. Revise widely. And make sure you prepare for statistical tests in that paper 3. Okay, guys, good luck. You're nearly there.